Hi, my name is Olivia Balkwill, and my summer research project asks which factors affect the level of mycorrhizal colonization in plant roots. One of the most common mutualisms are arbuscular mycorrhizal fungi, which are present in more than 70% of vascular plants. Since it's so common, I focused on these fungi for my project. The mycorrhizal fungi mutualism with plants is nutrient-based. This is because the fungi, which colonize plant roots, are highly branched and able to effectively obtain nutrients like nitrogen and phosphorus from the surrounding soil. The fungi then provide their plant host with these nutrients, which help the plants grow. This is why plants colonized by mycorrhizal fungi tend to have higher growth rates. In exchange for nutrients, plants provide the fungi with carbon. This is important because colonization represents the interaction strength between plants and fungi. My study asked the question, which factors influence the levels of mycorrhizal colonization in plant roots? Previous research has attempted to answer this question, and the result was the development of the passenger hypothesis, which postulates that variation in the characteristics of the surrounding plant community may cause variation in the level of fungal colonization in host plants. This can be subdivided into two characteristic types, abiotic and biotic. For the purposes of my study, the abiotic factor I focused on was soil nutrient content, and the biotic factor was the surrounding plant species diversity. My first hypothesis focused on soil nutrients. Research has shown that mycorrhizal fungi are more common when resources are scarce, so I hypothesized that lower soil nutrient availability would increase fungal colonization levels. My second hypothesis focused on plant diversity. Research has shown that more diverse plant communities have more fungal inoculum in the soil, which is why mixed plant cultures have higher levels of fungal colonization than monocultures. I therefore hypothesized that a greater surrounding plant diversity would cause higher levels of fungal colonization. The study site was a 40 by 40 meter plot in an old field at the University of Guelph's long-term mycorrhizal research site. Two focal species were analyzed, Hypericum caspitosum and Plantago lanceolata, and these were chosen because they're responsive to mycorrhizal fungi. The plot was divided into quadrats, and from each quadrat, soil cores were collected to determine the root colonization of the focal species and the soil nutrient levels, and the vegetation surveys were conducted to determine the above-ground plant diversity. The three dominant surrounding species were Poa compressa/pratensis, Solidago canadensis, and Bromus inermis. Poa compressa and pratensis values were average because they were hard to distinguish in the field. This graph represents the study site with the x and y axis representing the site's length and width. The z-axis shows the proportion of mycorrhizal fungi in each quadrat. Each data point represents one plant. Overall, this shows that there's variation in fungal colonization across the site. Although this graph only depicts one species, the others showed a similar pattern. There was no support for the nutrient hypothesis because none of the nutrients significantly affected fungal colonization levels, but there was support for the plant diversity hypothesis because the presence of one of the surrounding plant species, Poa compressa/pratensis, did affect colonization levels. These graphs compare the proportion of two fungal structures, arbuscules and vesicles, as seen on the y-axis, to the presence and proportion of Poa on the x-axis. As you can see, the presence of Poa differentially affected the fungal colonization in the two target species. More Poa was associated with more fungal colonization in Plantago, which can be seen on the left, and more Poa was associated with less fungal colonization in Hypericum, which can be seen on the right. Overall, there was support for the passenger hypothesis, but only in the context of the plant diversity hypothesis. This study found no significant effects of nutrients on fungal colonization. Since the presence of Poa did affect root colonization, the plant diversity hypothesis was supported. More broadly, these results suggest that at this particular site, biotic factors had more of an effect on fungal colonization than abiotic factors. Thanks for listening to my presentation.